Thanks so much again for the kind invitation. So my lab focuses on developing systems biology approaches to understand liver tumor biology by using single-cell spatial approaches. Today, I will talk about biomarkers of liver cancer from a single-cell view. As you all know, liver cancer comprises two clinical subtypes, hepatocellular carcinoma and intrahepatic cholangiocarcinoma, accounting for about 90% and 10% of liver cancer respectively. Both the two clinical subtypes of liver cancer are very heterogeneous, with vast molecular heterogeneity both between patients and within a specific patient. Those two layers of tumor heterogeneity make liver cancer one of the most difficult to treat malignancies and bring critical challenges to identify biomarkers for diagnosis, intervention, and prognosis. The question is, how do we better characterize the complex landscape in liver cancer, including the heterogeneous tumor cell population, the major cell types, the cell-cell interactions, as well as the potential spatial relationships of different cells to identify potential biomarkers. We applied the single-cell profiling to the tumor biopsies collected from HCC/ICC patients who were enrolled at NIH Clinical Center for Immunotherapy clinical trials. And we further used 10x genomics platform to perform single-cell transcriptomic profiling. This study was in collaboration with Dr. Tim Grayton and Dr. Jensen Hernandez from the National Cancer Institute. With the obtained single-cell profiles, we first separated malignant cells and non-malignant cells by inferring large-scale chromosome copy number variations. We found non-malignant cells were mainly grouped according to their cell types, as annotated using cell type-specific marker genes unique to T cells, B cells, tumor-associated macrophages, fibroblasts, and endothelial cells. In contrast, the malignant cells form the patient-specific cluster, indicated by using different colors here. This indicates the intertumor heterogeneity, means tumor heterogeneity between different patients. In addition to that, cells within a tumor differ in their transcriptomic profiles, showing evidence of intratumor heterogeneity. Then we are interested in the level of intratumor heterogeneity of each individual tumor sample. Is it a situation of very low degree of heterogeneity with one or two clones within each tumor, or high degree of heterogeneity with a very high mixed tumor cell population? And the following questions would be that, is the degree of tumor heterogeneity associated with patient prognosis, and whether surrounding stroma cells behave differently between low diversity and high diversity tumors? To determine the level of intratumor heterogeneity, we applied principal components analysis to the tumor cells in order to project the tumor cells to the eigenvector space to avoid noise and capture major information. Then diversity score was calculated by using the principal components. Tumor samples were divided into high diversity group and low diversity group using the median value. And remarkably, we found that the prognosis of the patients from the high diversity group has significantly poor overall survival than patients from the low diversity group. This was further validated by using an additional set of sequentially enrolled patients. These findings indicate that tumor cell biodiversity. We further studied intratumor heterogeneity by defining tumor cell states in order to reflect the tumor functional clonality. This is based on the concept that tumor cells are organized in a hierarchical manner. The algorithm of defined tumor functional clonality is mainly composed of 5 modules, including cell sampling to avoid unbalanced cell number, the use of consensus clustering to identify cells with similar transcriptome, the incorporation of stability test, the use of neighboring similarity search to identify to map cells to all corresponding clusters, as well as an iteration of the whole process for 100 times to identify stable tumor function clones. By using this strategy, we identify function clones within each tumor, and consistently we found that patients of tumors with a high clone number had a much shorter survival than patients with a lower function clone number. This further suggested a link of tumor functional clonality and patient prognosis. When looking at non-malignant cells, we found that non-malignant cells derived from high-diversity tumors, the red dots, and low-diversity tumors, the dark gray dots, differ in their transcriptomic profiles. And the difference is further evident when individual cell types was analyzed. Then we sought to determine if high-diversity tumor cells could produce some cellular factors that could induce tumor microenvironmental reprogramming. And we developed a strategy to search for cytokines or growth factors that were found upstream of at least 3 of the 4 cell types evaluated. And we further selected those that were differentially expressed in high diversity and low diversity tumors. By using this strategy, we found vascular endothelial growth factor A, VEGF-A, was a top candidate responsible for tumor microenvironmental reprogramming. And consistently, we found VEGF-A was mainly expressed in malignant cells rather than non-malignant cells. And we also found that VEGF-A is the expression is much higher in high-diversity tumors than low-diversity tumors. This was further validated by using immunohistochemistry analysis. And the increased expression of VEGF-A was in parallel with HIF-1 expression and hypoxia signaling. We also evaluated the VEGF-A downstream signaling as its function surrogate in non-malignant cells and found that the VEGF-A downstream signaling could discriminate endothelial cells, macrophage, fibroblasts, but not T cells. The role of VEGF in tumor angiogenesis has been very well studied. VEGF has multiple functions in the tumor microenvironment. Except for angiogenesis and vascular permeability, it may also act in an endocrine manner to promote dedifferentiation and EMT phenotype to enhance tumor cell invasion and survival. In addition, it may act function as a chemoattractant to recruit regulatory T cells to inhibit anti-tumor immune response, and may also recruit fibroblasts to affect tumor growth. Taken together, these studies demonstrate that high-diversity tumors may produce more VEGF, which may in turn reprogram the surrounding tumor microenvironment to affect the patient prognosis. Since tumor cells are so heterogeneous, we ask a question that, is there a stable molecular map or fingerprint underlying tumor biodiversity to reflect the intrinsic tumor biology? Intratumor heterogeneity may result from the evolution of tumor cells, but in addition to that, the continuous communications between tumor cells and tumor microenvironment may also contribute to intratumor heterogeneity. Stroma cells and immune cells in the tumor ecosystem may continuously communicate with tumor cells through cell adhesion, signal molecules, and small vesicles. Some of them may play an anti-tumor role, others may be reprogrammed or educated by tumor cells to promote the continuous survival and dissemination of malignancy. Therefore, define the molecular communication networks between tumor cells and tumor microenvironment may be a key to improve our understanding of the molecular map underlying tumor biodiversity of our tumor ecosystem. To understand whether the communication networks within each tumor are stable, we performed the single-cell transcriptomic profiling of liver tumor specimens collected from multiple locations of a resected tumor lesion. Specifically, we collected 3 regions from tumor core, 1 from tumor border, and 1 from adjacent normal tissue. With the collected tumor samples, we performed single-cell RNA sequencing to build a multi-regional single-cell landscape in liver cancer. And this study was in collaboration with Dr. Jens Marquardt from us in Germany. We determined the communications between tumor cells and tumor microenvironment by using a method called SampleDB, developed by Dr. Sarah Tuckman's lab. And the method allows inferring the ligand receptor interactions of malignant cells and the different types of non-malignant cells in the tumor microenvironment. In this dot plot, each dot represents a ligand receptor pair, The two colors, purple and green, represents the directions of the interactions, either from tumor cells to tumor microenvironment or from tumor microenvironment to tumor cells. And the size of each dot represents how stable the ligand receptor pair or the communication is. As you may notice, the majority of the dots are with a very large size, indicating that the communication networks are very stable within each individual patient. In addition, patient-specific communication networks are evident. For example, this group of communication networks only occur in this HCC patient. This was further revealed from this heat map with very stable but unique communication networks with each individual patient. To determine if patient-specific tumor and tumor microenvironment communication networks are biologically important, we extended our research for ligand receptor interactions in additional 24 tumor samples from HCC/ICC patients from the NIH Clinical Center. And we identified two major clusters based on the hierarchical relationship of the ligand receptor interaction activities. And noticeably, patients from the two clusters have significantly different overall survival, suggesting that the tumor and tumor microenvironment interactions networks are linked to tumor biology. Because this cohort includes both HCC and ICC patients, we also analyze HCC patients separately, where a consistent trend of survival difference between two clusters was found. To determine the key interactions of each individual cluster, determined by using the hierarchical relationship, we evaluated the difference between the proportions of each ligand receptor interaction in the two clusters. And noticeably, the ligand receptor interaction activities are polarized between the two clusters. For example, the LGALS9, SLC1E5, SPP1PTGR4, representing the communications between tumor cells and tumor-associated macrophages, were among the top of the interaction pairs from the cluster 1. And to further validate the robustness of the ligand receptor interactions linked to HCC prognosis, and we evaluated those two pairs between tumor cells and tumor-associated macrophages by using RNA-scope multiplex in situ hybridization as a kind of proof of principle analysis. Consistent with our single-cell analysis, we found that patients with high expression of both the pairs had a significantly worse overall survival compared with patients from the low expression group. Taken together, this multi-regional single-cell example suggests that there's a very stable but unique communication network between tumor cells and tumor microenvironment within each patient that underlies tumor biodiversity. And this network represents a very stable feature for HCC aggressiveness, opening a path for therapeutic exploration. We are also interested in biomarkers during liver cancer evolution. For example, in this hepatocellular carcinoma patient, we collected one tumor biopsy before immunotherapy and two other biopsies after this patient received the immunotherapy. And my fellow modeled the tumor functional clonality over time and to try to identify biomarkers during tumor evolution in response to immunotherapy. And we have a manuscript in preparation. In summary of the examples I have talked, tumor molecular diversity and tumor functional clonality are linked to patient outcome. Tumor-derived VEGF may drive tumor microenvironmental reprogramming, and the communications between tumor cells and tumor microenvironment may reflect the intrinsic tumor biology, specifically the communications between tumor cells and tumor-associated macrophages may represent tumor aggressiveness. We also have an online database called Single Cell Atlas in liver cancer for researchers to explore our single cell data set. Lastly, I would like to thank my lab members, my collaborators, as well as my colleagues, especially my mentor during my postdoc training, Dr. Xinwei Wang. And thank you for your attention. I'm happy to take questions later. Thank you so much.